It's my great pleasure today to welcome you all here, and we're going to hear from Dr. Motti Gerlic. Motti might tell you that I'm not the best person to introduce him because I have never read his CV. That may or may not be the case. However, I should say we did get an excellent reference from one of his colleagues who he played basketball with, who told us that he was a real team player, and that certainly has served him well over the last 3 years here at WEHI. And over that time, I've heard many stories from Motti, so I've sort of filled in the gaps of his CV. His time as a postdoc at the Sanford Burnham Institute, his time as a PhD student at the Ben Gurion University in Israel, and also as a paramedic in the Israeli army, and before that, his childhood on the farm. I think that there'll be a number more stories from this chapter in his life. It's been a terribly productive 3 years. In the last 3 years, he's published first author papers in PNAS, immunity, cell metabolism, and now he's going to tell you a last author paper that he's published recently, a co-last author paper in cells. So obviously that's a tremendous body of work, and based on that, he's been now awarded a faculty position at Tel Aviv University that he's moving back for in a few weeks. So you'll be sorely missed, Motti, but we're looking forward to many more years of collaboration. And your talk, which is intriguingly titled RIPK1, Goddess of Survival.
Yeah. Thanks, Ed. Can't work with microphones. First, I think this slide is more for Tracy asked me to put it, so please, all the students out there, go and register. So as I put my titles, and it's really coming from the big work that we just published in Cells, is really putting RIPK1 as one of the main players in regulation of cell death, either apoptosis or necroptosis. And in the case of necroptosis, what I will show you, it's also very important in inducing inflammation. Now, we are in the World Cup, and as I said, Seth maybe didn't read all my CV, but I'm kind of a mixed nationality. I was born in Argentina and raised in Israel. When it comes to football, and not soccer, sorry, it's football. I think I want to show you how I think the Argentinian people, at least that work on the cell death field, look at it. And apoptosis, where I am here, is, as we all know, is very regulated process, and it's taking time. You know, you get first signal from maybe TNF, you get some inhibition. You need to take out all the defender out, all CIPs and other inhibitors of apoptosis, and then you can really proceed. Now, this is a very long, I think, one of the best, I think, team effort. I can kind of jump because it's really long. You can see count of how many signal transduction is going on. But I think it's really, the result is of course amazing, although we lost later. Now, I will just move, and this is maybe even more famous, and that's how I look on necrosis. So we historically thought that, when you cell is trying to go to his apoptosis machinery, something is going wrong, it cannot really proceed, and that's how I think the field was looking on necrosis until 2003. You try to do it organized, something happened, defense kick it, some magic happened, and it's in. But really, in 2003, I think we started to understand that if you look actually closer, it's actually very much regulated, right? It's not a coincidence, and that's where I think necroptosis field really started. So after this World Cup, and I will get back to that later at the talk, I get into a more a background in the field, and really, as this project is focused on lethality of RIPK1 mouse, so I just want to introduce a little bit about embryonic lethality and cell death. So the RIP1 knockout mice were developed in 1998 by Michel Callar, published that on a mixed background. These mice are born, but they die a few days after birth. Now, in the same kind of time, 1998, 2000, and again 1998, also the caspase-8 knockout and the FADD knockout were developed, where in this mice, apoptosis, at least the extrinsic pathway is blocked, but these mice actually really die at early time at E10.5. Now, somehow surprising, when you take C-flip in 2000, which basically take the inhibitor of apoptosis, so now you actually induce apoptosis in these embryos, actually the mice die exactly at the same point, suggesting that, again, too much is not good, but also too less is not good. Now, as I said, the field in 2003 started where it's developed and really started to understand that there is actually another machinery, this necroptosis machinery. So really in the last 5 years, I think basically from all this paper, the field also continued to jump. And basically what they show that in the caspase-8, when you don't have caspase-8, it's not that you don't have cell death, it's actually that the cell death is switching into the necroptotic, and you can rescue it by taking RIPK1, which is an important, I would say, protein in the process. It's still unpublished, but this is a work here at WEHI, and this is in Strasser lab, and we know that MLKL can give the same protection. Same goes to FADD, protected by RIPK1, and when you look on C-flip, a little more complex, you first need to block the apoptotic, so you need to take FADD, but then you still need to come again with blocking necroptosis to actually rescue these mice. And a very complex slide that we kind of put together, and really what I want to show you here is that when you come to RIPK1, it's a very complex story, because RIPK1 is really known to play an important role in induction of NF-kappa-B, inflammatory cytokine, and induction of anti-apoptotic protein as C-flip. But it's also important in the complex that initiates apoptosis. And as I said, from 2003, we realized that it's actually playing an important role also in induction of necroptosis as the necrostatin 1, which is a specific inhibitor of the kinase activity of RIP was able to block this cell death. So as I said, the story which was published recently, really it's about RIP1 regulates the necroptotic pathway that will induce systemic inflammation. And this was really, if I will be short in time, I really want to say a big thank you. And really, most of the work was done by these 3 PhD students that all of them are finishing now. James, that started MD, Joseph, that is finished, and Joanne, that she's moving to the States for her postdoc position soon. So really, the question that we try to address is why these RIP1 knockout mice are dying. Now, just to say that at WEHI, after these mice were cleaned into the black 6 background, they did not even survive up to 3 days. They really died just at birth, and I would say a few minutes after birth, they will die. So what can we see in these mice when we did first some histology to understand what's going on? Look on a cell death parameter, in this case caspase-3 and caspase-8 cleavage. So caspase-3, we could see it in multiple organs, lung, liver, intestine, and thymus. And we also had, I would say, necrosis or necroptotic patches mainly in the liver and some in the thymus. More surprisingly was when we look on the plasma of these mice, and you don't get a lot of blood from a born mouse, but this is basically a 3 microliter of plasma in each well here, and you can really appreciate the amount of cleaved caspase-3 and cleaved caspase-8 in these RIP1 neonates. So really suggests that there is a lot of cell death going on in these mice. What we also saw when we analyzed the blood is that these mice seems to have a very, I would say, they have a systemic inflammation. When you look on the blood, very high levels of white blood cells, and they tend to be polymorphinuclear, or if you do a FACS stain, as I love to do and I love to do as well, they are mainly neutrophil. The cytokine profile in their blood was really, almost every cytokine was elevated, and I will just point out that in some, in correlation with the neutrophil, you can see that GCSF is really around 10 to the 5 fold elevated than their littermate control. this inflammation is not just in the peripheral blood. You can see it in different organs. And during the talk, I will really focus on the skin as the main organ that we can detect the inflammation. And you can see the hyperplasia of the keratinocyte. And even if you take lysate from the skin and run it on a bioplex, again, all the inflammatory cytokine and chemokine are elevated. So really just a quick summary on what we saw when we just really phenotypically analyzed these mice. What we saw is if you take RIP kinase 1 out of the system, you get apoptosis. We see cleaved caspase 3 and cleaved caspase 8. We also had necroptotic patches, so suggesting that there is necroptosis maybe in the same mice. And really what we see is a very severe inflammation in the mice, and they really die a few minutes after birth. So because we saw this severe inflammation, we were thinking that maybe the hematopoietic compartment, as usually, and again, neutrophils were very high, maybe that's what is causing this inflammation. Something is not correctly regulated in the hematopoietic that caused this inflammation. So what we try to do first is to see if we can transplant and transfer the inflammation. So you take fetal liver cell from E13.5 embryo and you transplant it to a wild-type mice, and you basically look if these mice develop any sign of inflammation. Now the quick question is no, we could not detect any inflammation. This is just bioplex, even in a very early time point. In this case, it's 2 weeks after transplant. But what we were able to see when we really tried to look on, and let's focus on Lys6G, that's a marker for neutrophil. Remember, RIP1, they have really high levels of neutrophils, so we were kind of hoping to see that these neutrophils are also building up in our transplant experiment. But really, the levels were really similar 8 weeks post-transplant, and really focus on the blue lines. That's the donor cells. That's really the wild-type ear and RIP1 ear, and really the same level. But what we noticed when we follow up these mice is that actually, as the title says, the contribution of the RIP1 cells decrease over time. So 14 weeks or 26 weeks, actually these cells are totally disappearing. You can see it also on the monocyte, T cells and B cells. They are really disappearing from the system. So that suggests that maybe there is, maybe these mature cells are dying. And to be sure if it's just a mature cells or maybe it's an early stage in progenitor cells, we also analyze the progenitor compartment. And you can actually see this is 8 weeks after transplant, that you already see this phenotype all over across the board of the different progenitor stages, suggesting that it's, initiated by already in the progenitor stage. Now to really make it short, we wanted to see if this is indeed intrinsic to the progenitor or there is something in their environment, maybe the mature cells are dying or secreting any cytokine that actually kill these progenitor cells. And for that, we did a competitive transplant when you take a 1-to-1 ratio of wild-type cells and RIP1. And if there is something in the environment that the cells secrete, this ratio will actually stay the same because both of them will die or proliferate the same. The short answer is that this is an intrinsic defect in the progenitor cells. And basically, if you do a 1-to-1 ratio, you see that the RIP1 cells cannot compete. And really, most of this work was done by Joanne O'Donnell, and she gave a full 45 minutes on the subject. So I will kind of stop here when it comes to the progenitor and the hematopoietic defect scenes in the RIP1 knockout. But really, when you think about it again, so I said that these mice don't develop any inflammation, but we have this progenitor defect. So going back, can we really claim that the hematopoietic is not playing a role or not initiating the inflammatory disease? So my answer is, I believe, yes, it's still standing by itself. And the reason is that you can do a different model, and this is a model that we did when we published in Immunity. This is an NLRP1 active mutation. Again, I have a very severe systemic inflammation in the old mouse. I have a clear defect, in this case, a paroptotic cell death in the progenitor cells. Very similar when you look in the inflammation and the progenitor defect. But when you do the transplant, although this, if you see here, that's the active mutation, they're really poorly reconstitute, we could clearly get this inflammation going on. So we should be able to actually detect the inflammatory environment or the inflammatory initiation if it's really the hematopoietic that initiating everything. And this is unpublished data, and using a different mice model. In this case, it's the SHP mutant mice, and I will not get into details, but what I want to show you is that when we try to rescue it again by crossing it to the RIP1, and this is again a transplant experiment, the Motti mice, as you can see, in 40 days, all of them are dying from inflammation. When we did the double knockout, you get some protection, but still, you can clearly see that you can transfer the inflammation even on a RIP1 knockout. Suggesting that on the RIP1, if you just move them, if it is the hematopoietic compartment, you should see the inflammation. And I will just mention this because I'm at WEHI. We have a Kanaganti group really use a very similar mice model with SHP mutation. She actually offered us to go back to back with our result because we have some protection on the RIP1. We said we don't think it is actually a RIP1 because we think it's a progenitor defect. She ignored us and published it in Nature. We will probably go back one day into that. So really, this part, I really want to say that we believe that the hematopoietic compartment by itself is not initiating the disease. It, of course, does not mean that it's not playing a role in the inflammation in the mice. So going back and thinking of why these mice are dying, and really the first thing that we want to focus is try to look on the apoptotic, I will say, scenario. So we have TNF maybe or Faz ligand that can initiate apoptosis. Maybe they are elevated in the mice and they actually started everything. And to be sure, we decided to use a knockout mice, which are knockout either to the receptor or the cytokine or to the downstream, the initiator caspase 8. So really using a genetic tool to ask the question if this arm is what initiating the lethality in the RIP1 mice. So we can see here the TNFR1 cross, TNF and Faz ligand knockout cross, and the caspase 8. Now you can see you don't really need to do even histology. You can clearly see that none of them protect. So you can see that this is a double knockout, again, triple knockout, a double knockout. And all of them still die at birth, and they're indistinguishable from the straight RIP1. Somehow surprising, because that's something that we were actually hoping to be able to see. We were hoping to see that the sign of apoptosis is disappearing from these mice, but the results are a little different. There is some decrease in the level of caspase 3 in the plasma of these mice, at least on the TNFR1, not really on the caspase 8, but it's still very clear in the plasma where it shouldn't be. And of course, wild type, you don't have any bands in plasma of the mice. So that was, I would say, somehow surprising that we didn't get protection there. What about the inflammation? Not surprisingly, I would say the inflammation was still going on in all this, I would say, double knockout mice, TNFR1, caspase 8. Again, if you look on the skin as well, one inflammatory tissue, you can see that it is not giving protection from the straight RIP1. And again, if you look on white blood cells and the cytokine profile, they're really giving you the same picture as the straight RIP1. So we conclude that at least at birth, and I want to stress that this is at birth, apoptosis is really not playing a role, or it's not responsible for this very early death. What we were able to see, and that's jumping back into the hematopoietic compartment, what we could actually see is that if we treat our transplant mice during the first 3 weeks post-transplant with anti-TNF antibody, we could actually rescue this engraftment defect. And this is a log scale. So, Devo, this is actually very significant. But we can rescue it. And again, when you come back, when you think that the TNF didn't rescue the lethality of the mice, with the result that we can actually rescue the hematopoietic compartment defect, but still these mice have no inflammatory disease, again, strengths that the hematopoietic by itself cannot initiate this lethality. So coming back, as I show you, at least by genetic tools, we could not rescue the mice by the apoptotic arm. So we decided to go and look on the necroptotic arm. Now I will just mention, so we decided to focus on RIP kinase 3 and MLKL. It was, I would say, a long shot in that time, because really what was believed in the field is that RIP kinase 1 is always required for necroptosis. There was one exception from Bill Kaiser group and Ed McCorsky that during MCMV infection, where MCMV itself can inhibit RIP kinase 1, you can actually go to necroptosis without RIP1. So that was maybe the only clue that necroptosis can be a RIP1 independent. So we did the crosses into the RIP kinase 3 and into the MLKL, and you can really see this is just after birth. You really need to go and do genotype to really say which mice is really a RIP1 knockout mice. They were really, and we could not see any sign of inflammation, or at least not by the eye. We were maybe not so happy to see that after a few days, usually between day 3 to 5, these mice actually never gained really any weight, and they basically died a few days after. So it wasn't a full protection, although at birth it seems to be a full protection. So when we go and look on these mice, and I'm just showing you some of the data, and in here it's a caspase-3, so I just want to, without showing you the data, I will just say that in most of the organs that we were looking, there was a significant decrease in cell death when you look on the lung and thymus and etc., except of, I will say, the intestine. So if you look on the small or the large intestine, at day 0, you see some caspase-3 activity, and really at day 3, in these double knockouts that now can survive to day 3, this is actually getting even worse, mainly in the large intestine, as you can see here. And just to reassure you, and that was again a surprise, that we could not detect any sign of caspase, cleavage caspase in the plasma of these mice. And remember, when we took out the apoptotic, either TNF or caspase-8, we still had very clear bands of cleaved caspase-3 in the plasma. So this was a surprise, and this is probably a still unanswered question, I will say, in our experiment model. Why, when you take necroptosis out, you really don't have a caspase-3 cleavage, where you take the apoptotic, you still have it. Inflammation in these mice, the double knockout mice, I would say RIP1, RIP3, and MLKL, everything is restored to normal values. As I said, at birth, and this is even at day 3, they don't develop any inflammatory disease. This is the skin at day 3 again, except of not gaining weight. They seems to be normal. So the only exception with the cytokine is really MCP1. But even this one, the level here, which are around 5,000, are at least 10-fold, if not even 100-fold less than the real RIP1, which is much, much higher. So all the inflammatory phenotype really disappear when we took necroptosis. Again, if we go back into the engraftment, and this is not a finished story, I will say, and we did the first transplant using all this, of course, genotype, try to see if really necroptosis is driving the engraftment defect. And really, if you just know too many colors, if you really focus on the purple one, which is the double knockout, and we can even focus just on the LSK, which is the progenitor compartment, you can see that you have a defect in the RIP1, but actually if you take RIP3 out of the equation, it seems to be normal. So it seems to be that necroptosis is important to initiate this engraftment failure or defect. But when we push the system, and usually you actually want to push the system, you do a serial transplant. So when we take this bone marrow from these mice and transplant it to another recipient, and then come again after 8 weeks and analyze, then again, let's focus just on the LSK, you start to see a decrease. It's still higher than the RIP1. They are doing better, and again, that's a log scale, but you actually start to see a phenotype here. So that's, right now we are in the stage that, we are not, I will say that probably necroptosis and apoptosis, both of them are playing a role, and that will probably not be surprising because we don't really talk here about homogeneous population when we actually talk about transplant, and so that can be actually the different. And if you really start to focus, and we didn't follow up, B cells, for example, seems to be still normal, where T cells are maybe much more affected. But this is probably something to follow up in the future. So really, to summarize the genetic evidence I show you so far, when we take RIP1 out of the system, out of the animal, we get these two arms. We get the apoptotic and necroptotic arm. Now, if we delete the apoptotic arm, which is in this side, we still have the necroptotic, and the mice are still dying exactly the same as the RIP1. There is no real difference between them. They still have a severe inflammation. But when you take RIP3 and MLKL, the necroptotic pathway, they die at day 4, they don't have any inflammation, and that's the reason kind of switch it, that basically what this suggests is that the inflammation is a secondary event for the necroptosis cell death. So really, we decided to try and really focus on this inflammation that seems to be very crucial to the lethality of the mice, and really ask what is causing this inflammation. Now, in the necroptotic field, there are plenty of danger-associated molecule that suggested to induce this inflammatory response. The problem is that most of the work is using cell culture, ex vivo, and not too much was done in vivo. So it's really, although we have kind of a list of suggested dump, we didn't, have any really clear clue which one can really play a role. So that was kind of more fishing and depend on the tools, of course. I will just mention ATP, for example, which, if it is released from necroptotic cells, will induce inflammasome activation. I'm not going to show you any data on that, but we don't think the inflammasome is playing any role in the lethality of these mice, and I will just skip this one just because of time. Mitochondrial DNA, I will say that we could not detect it, but I think there is some technical issues there, so I will not want to claim that it's not playing a role here. What we were able to really at least detect using a Western blot is HMGB1, IL-33, and IL-1 alpha. And what we basically saw is, if you look on the plasma of these neonates, RIP1 and the littermate control, when you look on HMGB1, and in here we have a positive control, again, ex vivo, very nice blot, nice band, the antibody works good. When you come to the plasma of the mice, we were actually first surprised that we can actually detect the same specific band in our control. So what is actually telling us, I'm not sure. And in the RIP1, we can, I would say, detect the same levels of specific band, suggesting that at least in the RIP1 setup, HMGB1 is probably not a major dump. What we were able to see on the, and I'll show you down here again, is IL-1 alpha. So IL-1 alpha, we could not detect it in our control plasma. We could detect it in the RIP1. And I will just say that this is not ex vivo, this is every lane ear or ear, it's a different mouse. So this is 4 mouse here, 4 mouse here. And very clear you can detect it and not detect it in the control. And I will just want to mention that the idea behind IL-1 alpha is that it was suggested that IL-1 alpha is actually in the nucleus, and during apoptosis was shown to bind very strongly to the chromatin, suggesting that during apoptosis it's really not getting released unless the cells are not getting engulfed and at the end everything. But even then, it will be together with the chromatin. where in the necroptosis, as you can see here, it was suggested ex vivo, but we can actually see some evidence in vivo that it's indeed getting released. The more strong evidence, I think, came from the IL-33. It's another IL-1 family member, and was suggested to be a necroptotic dump. And really what you can see here is, if we focus here, RIP1, really high amount of IL-33 in the plasma. We do RIP1 caspase 8, which of course doesn't rescue the inflammation. Again, you still have this very nice and big band. In our necroptotic crosses, this band is really totally disappear. So I think that was our best candidate, I would say, as a necroptotic dump. And that's something that we will follow up by crossing our RIP1 into the IL-33 knockouts if there is any protection there. So as I said, we have a candidate which is IL-33 and IL-1 alpha, but luckily for us, if you actually look on this pathway, what is interesting that actually almost except of ATP, which is the inflammasome, all of the other dumps, all of them need the adapter MYD88 to signal and to cause inflammation. So that actually gave us an opportunity to go back into the genetic tools and really see, can we delete MYD88 from the RIP1 and get any advantage? And the answer is yes. The MYD88 RIP1 double knockout mice in the left ear really seems to be totally normal at birth. I will just say they are almost as good as the RIP1, RIP3, and RIP1 MLKL. Again, they die around day 3 to 5. They don't really have significant inflammation, maybe a little bit elevated white blood cells, but really cytokine profile, again, except of MCP1, again, seems to be normal. If we look on the skin as the major inflamed tissue, again, back to normal. So that's really the only, as is one of the striking again, and we don't really have a good explanation. Again, in the RIP1 MYD88, we could still detect caspase, cleave caspase 3 in the plasma. I just want to mention that even, it's different mice, 1, 2, I will actually say that in this number 3, there is a band. It's a faint band, but there is a band where we really never be able to detect any bands on the RIP1, RIP3. So it seems to be that maybe the RIP1 MYD88 is somewhere intermediate between these samples, but it is something that we can detect. And again, that's something that we need to follow up to really fully understand why when we take necroptosis, we lose this cleave caspase 3. It doesn't really sit as with the dogma about necroptosis being a caspase independent, although caspase independent doesn't mean that caspase cannot get cleaved. So if we summarize and add the RIP1 MYD8 into this side, right, we basically add it here, blocking inflammation, we switch into this side, which is a P4. So the question is why these mice die. And if you look on lethality of mice around this age, one of the first things you ask yourself, can they actually get any food? Maybe they don't even, they can't even get milk. That's actually a story that Don was telling me that in one of his mice, that's what they actually found, that the mice could not suck. So if we look on these mice, and we can see it by histology, but I think this picture is even nicer. This is the RIP1 MLKL, plenty of milk in his stomach. There's no problem about getting the food into the system. What about bacteria? Maybe, these mice getting into the environment, you get colonized by bacteria in your skin, in your intestinal. Maybe that's something that is actually causing these mice to die, although we couldn't see any sign of inflammation. We still went further and put our RIP1, RIP3 into the gem-free facility, try to see if it can give any advantage to the mice. And just to summarize an expensive experiment, the answer is no. There is no evidence that if you do it on an abiotic environment, you get any significant rescue. So that takes me back into maybe the background slide when we look on embryonic lethality and cell death. And interesting, in 2012, the C-flip mice, as I said, die at E10.5. But when they develop a C-flip conditional in the intestinal epithelial cells, this mice actually is born and is actually dying around day 3-4. So again, this is a very clear apoptotic phenotype. You lose C-flip. If you have apoptosis in your intestine, that's kind of the checkpoint. If you take TNF, so they show it's a TNF-driven phenotype, you rescue these mice. So that's kind of took us back into our result. We don't really need to do any new experiment, but really analyze, be more specific, and look for clues for this apoptotic phenotype. And remember, I told you that the only organ that was still had a clear cell death phenotype in our RIP1, RIP3, RIP1 MLKL, and the RIP1 MYD88 was the intestine, either at day 0, which is, you can see it in the small, but really mainly in the large intestine, as you can see here. And you can see what happened at day 3. Again, in the large intestine, there is a massive accumulation of positive caspase 3 cells, cleave caspase 3 cells. And I will say even more, something that we didn't really follow up, but when we reanalyze our mice, what we can see in the large intestine, I'm just giving you the example from the RIP1 MYD8, which are basically the same as the RIP1 or the RIP1 TNFR1, you can actually see that there is a development defect in the large intestine. You're really missing all this layer of goblet cells. It's just not there. This is at day 0. TNFR1 seems to be giving some protection for this development. I didn't put here the slide, but actually RIP1 caspase 8, when we went back and tried to look on this development defect, I will actually say that this is almost fully rescued on the RIP1 caspase 8. It seems to be very normal, the large intestine. So taken together, I think what it's showing us that, and again, I put this question mark, I have no idea, maybe there is some crosstalk between RIP3 MLKL and caspase 3, and we can leave it there. But what I show is that really this RIP1, RIP3, RIP1 MLK, and RIP1 MID8, the reason that we believe they are dying at day 4 at this checkpoint is really apoptosis in their, mainly in their large intestine. So if we take this crosses together with this crosses, we actually got to the point that maybe we can finally rescue the mice. So if we take this mice and now take out caspase 8, maybe we can get a viable mice. And this is really what we got when we did a triple knockout mice, RIP1, RIP3, MLKL. Mice seems to be totally normal. This is a quick summary, just, you don't see the phenotype in the large intestine. Of course, you don't see any cleave caspase 3 because that's already blocked by the RIP1, RIP3. And in here, I'm just not going to focus on it, but these mice behave basically almost like a RIP3 caspase 8 mice, which develop a lymphoproliferative disease, seen here. And the only thing that you see is that the kinetic is a little bit slowed down if you take one allele of RIP1, and it's even getting even slower when you took 2 alleles. Just suggesting RIP1 playing a role also in this disease, of course. So try to make a simple summary from all this data. Really, what we think now is really, what we think is that RIP1 is really essential for controlling cell death. It's really on normal condition, it is part of a complex that can block either apoptosis and necroptosis. Now, if you take RIP1, although it was suggested to be very important for necroptosis, we actually think that if you take RIP1, you actually induce a very severe necroptotic phenotype, which can be initiated without RIP1. Don't really need RIP1 to do that. So that's, I think, one of the main findings, and maybe somehow a switch in the field. We also, one of the first, I think, stories to really try to address what is a necroptotic dump in vivo and see the sign of it, and try to even block it by, going downstream and look for the adaptor. And of course, there is still a big gap here that we need to fill out. If it's really IL-33, as I said, we should be able to rescue it by crossing it to the receptor knockout or the cytokine knockout. And again, this shows us, again, the specificity of apoptosis and necroptosis between different tissue. The intestine seems to be, at least when you come to RIP1, RIP1 is really controlling the apoptosis in the intestine, where in the skin, at least, it seems that RIP1 is controlling the necroptotic pathway. And as I said, the hematopoietic compartment, it's probably something in between. Now, going back into some World Cup, I think. So, as I said, I put necroptosis as the hand of God. But basically, I think what I showed you is that you don't really need this hand. And actually, this hand is maybe inhibiting. Usually, if you will use your hand, you get a red card and you don't score. But I think this is just to show that you still don't need, your full, I would say, apoptotic scenario. You don't maybe need so many teammates. You can still do it by yourself. Just don't use your hand. Now, for Devo, I think our results are standing. And again, it's a WEHI always issue, right? You want to know that you didn't do an artifact. So it's always good if someone else can repeat. And I think in this video, you can see that indeed you don't need RIP1. You can really reproduce it if it will work. A one-to-one. It's different time, different labs, but you still get the same result. Really amazing. I still hope that 2014 will finish as 86, but that's a different issue. And really, this takes me to, I want to acknowledge actually two groups, Dillon and basically Douglas Green and Bill Kaiser and Ed Mikorsky, because we really got to the point that we rescued the mice probably 1 year ago, and we met at Keystone. Everybody was excited. We actually share our main result that we can rescue the RIP1 mice. So all of us knew that each lab actually rescued the mice. I think we took probably a very smart move, saying that we will continue to talk about, try to publish together, but we don't share any results. So basically, everyone go independently on analyzing these mice. And it turned out to be really good because actually you get a very broad analysis, as I think we are very different from these two groups that took a very similar approach. But I think the overall picture is actually is a much more informative. So Green was publishing, we published with Green back-to-back in Cell, and really on the same week, Kaiser and Ed Mikorsky published it in PNAS. Now, without getting into too many details, I would just say that their hypothesis, when you don't have RIP1, and you can see it very clear here, and maybe this is a more simple, both of them claim that probably there is a signal too. I would say Green was maybe took it, he didn't really want to claim something specific, so he put a TLR3 interferon, where Kaiser really put interferon signaling here as a signal to which induced the necroptosis arm. So they claim TNF, both of them claim TNF make apoptosis, which we definitely agree when you look on the intestinal. I'm not sure that's the case when you look on hematopoietic. And they claim that interferon is what inducing necroptosis. Now, I just want to mention that all their data about interferon is an ex vivo experiment, with RIP1 knockout cells, although they actually, at least Green actually did the cross. He can do whatever cross he wants. So he actually generated a RIP1, TNFR1, IFNA cross. They were not protected. So again, if we definitely agree that ex vivo, you have cells that when you don't have RIP1, will go to the necroptotic pathway upon interferon signaling. I'm not sure this is actually the case when you look on the all animal and on the RIP1 knockout mice. So again, coming to this, I think, schematic, which kind of, I think, how the field was maybe before our paper. That's the field after our paper, but I want to say that we didn't contribute all of the, we contribute probably the main one is this RIP1 that basically can go directly into RIP3 and kill. But there are other papers really using a kinase dead RIP1 or kinase dead RIP3. And over there, it's even more of a mix, as some of them switch back into an apoptotic cell death, which I'm not sure how much is really true, or this is maybe just a consequences of some unfolded protein. And for me, what still is unanswered, or the main unanswered question, I think, in the field for me is still, if I think about RIP kinase 1, we still don't really understand how the same molecule is really initiating and inhibiting the same process. We know that the kinase activity is important for necroptosis, but actually from this set of data, you can see that you go to necroptosis without RIP1 at all, suggesting that maybe actually the kinase activity of RIP1, maybe the substrate for this kinase activity is something that is inhibiting necroptosis, and when you have a kinase activity, you take the inhibitor off, and actually you can go. So if you don't have this, that's one way to think about it, but I think there is a lot of experiment that we need to do to really answer this question. Second one, and didn't talk about that, but and Murphy is definitely, James Murphy is talk about that and is really strongly working on try to understand is MLKL is the end point of necroptosis, as if I took here. We know that now from MLKL it's getting into the membrane, but we don't really know if this is sufficient to cause pore and kill. Maybe there is something in between, maybe there is some another protein that it needs to bind. So I think this is something that really need to be clarified. Is this is an endpoint of we can do maybe something in between as well. And the more big picture is what is the physiological relevance of necroptosis? That's something that always people in the field like to ask. That's kind of a big question, so why it's important. We definitely have, we showed you that necroptosis is important here in the skin, and I would just say that James Rickard in his PhD, which work on the sharping mice, which is a kind of a model of skin disease. Again, it's a model. Again, necroptosis was the main driver of this, and he gave a full 45 minutes on that. And I think John mentioned it 2 weeks ago as well. So probably it's depend on the tissue, and we really need to find out which tissues are more prone to necroptosis phenotype. And I will just mention, and this is from, it's kind of behind the ear, it's probably playing a significant role during infection, where you can imagine that viruses and bacteria evolve to inhibit all these machineries, but sometimes they inhibit one machinery. For example, the MCMV, it can inhibit cell death by antagonized for C-flip. It has a C-flip homolog, a viral C-flip. It can also inhibit RIP kinase 1, but still the system in this case is going to a necroptosis cell death, which is a RIP1 independent. So it's another, I think, example of where necroptosis can play a role, and we really need to see in which infection it is important. And I think we started to touch it a little bit, but I think this is something that still we need to work on, really try to understand if they are specific dump for necroptosis, and if they are, try to correlate them into the physiological relevance. So maybe they can be a sometimes better target or easier target, I would say, than the molecules that are initiating necroptosis. And thank you, slide. I was a little lazy of copying all the names, to be honest. Really, again, big, I think, James, Joseph, and Joanne need to take most of the credit, I think. Here's Joseph, here's Joanne. And as I said, James is in Geelong, I think? No, it's in Monash doing his MD now. It's WEHI, so you cannot finish without really say thank you to the animal facility, and you can just imagine how much work the animal facility did here. Flow cytometry, hours of sorting for all the engraftment experiments, histology, again, a huge amount of work. Now, as it said, I'm actually leaving and this is just one project that I really work on. So I also want to really say thank you for all the collaborators, and I'm sure they know who I'm talking about on all the project that I was involved in the last 3 years. And again, if I come back, I say thank you to actually James, Joseph, and Joanne. On this project, I still want to say even thank you to John Silk and Ben Crocker that allow me to play a significant role in this study. I also want to say thank you for, it's a different thank you for Ben and Seth that they actually gave me the opportunity to come here and work at WEHI. And I think, especially I will want to say thank you to Seth because I think it's when I'm thinking about it, I'm going to open my lab. Sometimes it's not that easy to have someone and to give him so much freedom in your lab and especially when you start. And I think Seth, you need to get the credit for that, and I think that's something that I thank you, Seth, for that. And with that, I will just finish. If you go to the Middle East, I know it sounds like crazy out there. One fact that I actually learned is I thought that we are actually the biggest area of camels, as my wife, grandpa, was the most famous camel vet in the world. That's a fact. But I was actually surprised to learn that Australia is the biggest place of camels in the world. Don't know how they came here. I'm sure it's not, they don't have a pocket. So really, if you go into Israel, you are more than welcome to come and say hi. I'm getting upgrade from a 3rd floor that you don't have a view. I'm going up to an 8th floor, so hopefully I will be able to see the water from there. And really, I'm sure I will continue to collaborate with WEHI in the future. So thank you.
So the IL-33, you had it coming from a necrotic cell and then going to another cell and then causing necrosis. So where does it start? Who's the chicken and who's the egg?
Oh no, so maybe I did something wrong. It's me, I'll take the responsibility. No, so IL-33 is really, here we go. It is coming from the necrotic cell. So it's getting released. What I'm saying is that this is, a cytokine that will influence neighboring cell to an MYDH8 to cause inflammation. It will not cause death again.
So it doesn't cause...
No. It is just inflammatory cytokine.
Can we go back a couple of slides to this?
Yes.
Spaghetti one.
Wait, which one?
Well, one of the ones with lines.
Oh, the spaghetti one. This one, or this one. This one, here we go.
There was one where you had up in the corner, you had IL-33 signaling into the cell.
Yeah. So if you look at it again, I know. I'm following you, David. To be honest, it was 2 slides together. I tried to make it more simple, so I make it 2 slides. So if you go to the one that you're talking here, and if you see, this is actually I put here in the corner. This is a necrotic cell, and you have MLKL at the corner, and dumps are getting released. So what all these dumps are doing, they're causing inflammation. They don't cause a cell death. This is not a cell death. This is a...
So the big cell there, that's not a necrotic cell?
No, this is the necrotic cells. This is macrophage, T cells, whatever is sensing the dumps in the environment.
I have 2 questions. So what's the source of inflammation? And if you don't have RIPK1, RIPK3 in the immune source, would it become more inflammatory or less inflammatory?
Okay. So the first question, in the RIP1 knockout, what I think, and at least the genetic evidence that we have, is that the cause of inflammation is necroptotic cells. If you rescue the necroptotic phenotype, you don't have necroptotic, you don't have inflammation. Okay, so this is kind of this slide. You need this initiator here, which is because you lose this break, RIP1 is not able to stop necroptosis. I think it's a more spontaneous than maybe what Green think, that there is maybe interferon or whatever, but time will say if there is actually something that really, what E15 is out there in the embryo that start it. I actually think that this, I didn't show data, but I think there is more balance between the different proteins. So you don't have RIP1, you get maybe accumulation, and I didn't show of RIP3, and that's what actually causing RIP3 to maybe oligomerize and initiate necroptosis. So that's for the first answer. This is what initiates everything in this mice is necroptotic cell F. What happens if we take RIP1 and RIP3 out of the system? I think this is that's a 100 million question. I think you have so many labs try to figure it out. And I show in the slide, I didn't even focus on RIP3, but I can put it here, right? I said RIP3 is a necroptotic, right? But you can see here, and James Vince is working really strongly on this point. You can actually get RIP3 to induce inflammasome activation. So now, what's going on if you take RIP3 out of the system? Is it really influencing cytokine production? I think this is again coming to different cell types, probably, and then it's depending. If you work on macrophage, you get maybe one phenotype. If you work on dendritic cells, maybe it's a little different. So I think this is a much harder question, and I don't have an answer for that right now.
Time for one more question, I guess.
So in the mice that have no RIP1 and there's no necroptosis because MLKL or RIPK3 is gone, you showed a lot of caspase 8, cleaved caspase 8 in the plasma. Are you sure that's active caspase 8? Because just because it's cleaved doesn't mean it's active.
So you talk about the double ones or the straight one?
Knockout, yeah.
The double. So the double, we didn't see caspase 8, we saw caspase 3. So it's caspase 3 there. And I agree that we don't know for sure. I will just say that we did one attempt to look on activity in the plasma. Unfortunately, James was missing a few controls there. So I cannot say definitely, but it seems that this caspase 3 is active. But need to be repeated a few more times with the right controls to really say that. But yeah, it can be that it's just a cleaved product because the cells just die, and there is plenty of other proteases that are maybe going on, and this is just something that's artifact. I agree.
I think if you want to hear any more about camels or anything else, you can probably ask Motti later on, but we should thank you for a wonderful talk.